I love that for all the sophistication of the Simons Foundation, you get an analog pointer. Being reminded of Sandy, I have to say, if you go look up the article, what I really wanted to title it, partly because Canada's bilingual and partly because I thought it was cute, was Moi après la déluge, which is a play on Napoleon. Since the Simons Foundation is such a mix of mathematicians, physicists, and biologists, I thought I'd put it in perspective by telling you what I think is both one of the best and most apropos jokes about the three of them. So as the story goes, there is a biologist, a mathematician, and a physicist riding on a train heading up to Edinburgh, and they look out onto a field, and there, sure enough, as you might expect, is a sheep. And the biologist looks out and declares for all the world to hear that all sheep are white. The physicist, realizing that was perhaps too broad a statement, looks out the same window and corrects the biologist by saying, of the set of all sheep, there exists one sheep that is white. The mathematician, sadly shaking his head, you'll like this, Jim, looks out and says, of the set of all sheep, there exists one sheep that is white, at least on one side. So that really kind of speaks to the precision of biologists. But hey, you know, we're generalists, and that's where we live. So I'm going to talk to you about the subject that is near and dear to my heart for anyone who's on the younger side, and I think we have a few of them in the second row here. I have to say, if you enjoy science and you enjoy research, nothing is better than finding something that you really can put your heart into and love. And I have to say, I was lucky enough about a decade ago, prompted by my colleagues like Yuri Bajaki, to turn my sights on interneurons and their development. And I have to say, it's been wonderful and fun, and that's all it takes. You know, you can worry about how to start a career, find a question you love, and the rest is easy. Well, except for grant writing. So I was really disappointed when Nenad Sestin and Matt State started doing profiles of human brain development and concluding what neurons were important, because they, I think, settled more or less during development on the subplate cells. I still think they're wrong. And I have this fellow to support me, Ramon Cajal. This really doesn't work, by the way. You have to use the analog. So Ramon Cajal, among his many, many statements, made this as one of them, which is the functional superiority of the human brain is intimately linked up with the prodigious abundance and unaccustomed wealth of forms of the so-called neurons with short axons. It's a little verbose, so let's simplify that. He was talking about interneurons. So really, an individual who had looked across many species, from rabbits up to humans, compared the anatomy of them, looked at the many differences, in the end finally concluded that the chief difference that makes us special are interneurons. Now, the jury's still out on whether that is the sole and most interesting part. I'm certainly an advocate that it's an important part. But the idea really was that in humans, we have both more and more elaborate interneurons, and that has something to do with the ability of us to have a higher cognitive state than perhaps rabbits. So let's take a look at the cortex, and here's a Golgi stain where you can see the beautiful network of cells that come together to form it. And then if you pull it apart and look at the individual cells, you can see they're really formed from two major classes. Although this is much simpler and much more uniform in types, the so-called principal glutamatergic cell forms 70 to 80% of the neurons in our brain, and the remaining 30% are this ragtag group of interneurons that vary in abundance from maybe 2 or 3% down to sub 1%. So for the way I translate this into brain function is that they really share in the way our brain delivers the world to us. The principal cells are responsible for taking an external world and topographically reorganizing it in our brains, and the interneurons, by sculpting it through inhibition, is allowing us to attend and to coordinate information so that we can perceive and act on that world. So you can see that I've kind of slanted the discussion to saying this is about as interesting as a projector, and this is doing all the real thinking in the brain. I don't know if that's true, but it's certainly true that the interneurons, by having really, really a high degree of diversity, are able to have interesting computational tricks that clearly play on consciousness. So just to give you kind of a quick entry point for those of you who may be less anatomically oriented, the isocortex or the neocortex is called iso because it's very similar in all its parts. And that's because information flows through it in a canonical way, independent of whether you're looking at the motor, the visual, or the prefrontal cortex. It's 6-layered from inside to out, and information travels to it from subcortical areas, mainly the thalamus, into layer 4. It's rooted up into the superficial layers where it communicates with all the other brain regions. The information ultimately comes back to layer 2, 3, down to 5, 6, and goes out. And independent of where you look in the cortex, that's the basic organization. Now, at each of those junctures are sparse numbers of interneurons that shunt input, output, or throughput, or perhaps most interestingly, coordinate the rhythms and activities of the cells so that that activity is well-ordered and well-directed. So the flip side of normal brain function is, of course, a brain dysfunction. And that's beautifully represented from the series of paints from Munch, this one being one of the Scream series. And really, what the game in neurobiology in terms of psychiatric disease has been for the last 10 years has really been a game of trying to find out what genes, when mutated, cause psychiatric disease. And no group has probably committed more effort and money to that than the Simons Foundation. And the fruit of that is that when I started being a Simons Fellow some decade ago, I have to say, really the knowledge of genetics in autism came down to a simple fact. Monozygotic twins had a high concurrence rate. So we knew it was genetic, but frankly, all the data out there in terms of what genes were important was at best anecdotal. And an effort from numerous groups, most recently Evan Eichler, Matt State, Dan Geschwin, and others have done, and Michael Wigler have done a wonderful job, I think most powerfully using exome sequencing and really detailed private mutation analysis to identify genes that are really and truly associated with autism. The question is, what do you do with that information? How do you take that information and turn it into a real understanding of what is dysfunctional of the brain? So my own effort to do that recently actually was just today accepted at Neuron. I am the junior partner from Vermont of a paper authored by two people in the audience, and that would be Caitlin Mullins and Dick Chen, my partner in crime. And essentially, it puts forth the proposition that you have to think of the nervous system as a control system. And so here, kind of making the analogy with a HVAC system, you can think of neuronal firing as being regulated up and down. The thermostat on the wall is mediated through calcium signaling, and that leads to signaling to the nucleus, which changes transcription, creates a feedback loop that adjusts synaptic strengths and weights, and ultimately brings the system back into homeostasis. So this is really extending on work of others like Gina Torreggiano, who have made arguments about this. But I give Dick and Caitlin a lot of credit for this. In a systematic way, they really laid out and kind of allowed me to share in this a kind of a vision of how you might turn a control system like that, apply it to the nervous system, and think of how the genes we're discovering in autism might play out in terms of such an HVAC control system. And the key, of course, is the thermostat, the homeostat, which controls it. It leads to effector events through nuclear signaling and through dendritic events. This really plays itself out through protein signaling and splice variants, something I'll talk about in my talk today. And then ultimately, you either adjust the activity or the synaptic strength to bring the system back into balance. And if you start looking at some of the genes, like the L-type calcium channel, RB-FOX, FMR1, and others that are mentioned here, and many, many more that the Simons Foundation has described, you can begin to think in a holistic way how autism might be brought together, not as a single disorder, but as a single control system. So I really say that I don't know how right or wrong we are. Time will tell. It's really just a hypothesis. But what I can tell you, it was an absolute pleasure to work with Caitlin and Dick on this. And really, we scratched many a Saturday morning our heads trying to figure out whether this made sense or not. So soon you'll have a chance to tell us how we were wrong, but hopefully useful. Excitatory and inhibitory. So there's a lot of ideas, starting with Merznick and John Rubinstein and others. Actually, Michael Shadlin originally suggested this back with Bill Newsom, that the brain has this excitatory to inhibitory balance. And then when that gets out of sync, that's when things like autism occur. So this is a picture done by Robin Tremblay from Bernardo Rudi's lab. It shows an overlay of the interneurons as they weave their way from the layer 6 up to layer 1 of the cortex. And what we have done over the years and continue to do is to purify our favorite cell types, the inhibitory cells, and start probing using RNA-seq and now more recently single-cell RNA-seq with Rahul Satija down at the New York Genome Center to really understand the molecular profile of these individual cells. And kind of a back-of-the-envelope calculation, and like all back-of-the-envelope calculations, it shouldn't be taken too seriously. But when we took all the genes we've purified, identified as enriched in interneurons, and compared it to the vast list of genes that have been at least associated with a psychiatric disease, we got an overlap of about 6%. So is that high? Is that low? It's really hard to say. But when we compared that to the overlap in principal cells, it was about double. So I don't know if that's a meaningful fact or not, but it certainly does point to the idea, which we believe, which is that one of the reasons psychiatric disease occurs is because genes that function importantly in interneurons, when they dysfunction, have their impact because the interneurons aren't playing their role in brain function. So, in my world, yes. Not, you certainly can talk about interneurons in other places as literally what it sounds like, short conduits that aren't. But in the cerebral cortex, other than the stellate cells, which sit in layer 4 and are considered interneurons as well, they're pretty much a large class of GABAergic inhibitory cell, yes. So here are some, I think, bona fide examples of syndromic and non-syndromic genes that have clear relationships to psychiatric disease, autism, epilepsy, and intellectual disability. And these are genes that are clearly enriched in interneurons. Here's another view of it. And again, I offer this more as a bait to think about it in a certain way, but I have to say I find it very provocative. So one of the things I'll show you is that we've mapped out the origins of these interneurons, and really all the interneurons anywhere in what I would consider your brain, your cortex, your hippocampus, your amygdala, your basal ganglia, come from one of two structures. This structure, MGE, the medial ganglionic eminence, and this structure, the caudal ganglionic eminence. So these are proliferative zones that exist transiently in the embryo in both mice and humans. And having done a whole lot of work over a decade, I can say with confidence that every inhibitory cell in your brain comes from one of those two structures. And they migrate through this Baroque long-range migration to take up residence up in the cerebral cortex and the hippocampus, anywhere they're needed, and adapt beautifully to those circumstances. So in terms of the cortex and their integration, one of the things I think is an interesting thing to point out from an engineering point of view is how the cortex as a structure is built. So we all know from the first neuroanatomy class that the cortex is made inside out. So if you're not familiar with that fact, the deep layer cells are born before the superficial cells. But if you think of the structure of the cerebral cortex, it's really, in an engineering sense, made with an output layer made first, an input layer made next, and finally you make an associational layer on top of it, which connects those two input-output layers together to give function. That's the developmental ordering of things. If you look at the interneurons, the 70% of the interneurons that come from this structure, the medial ganglionic eminence, predominantly reside in the deep layers. So they're the major stakeholders that control input and output of the cortex, whereas the ones that come from the caudal ganglionic eminence are the major stakeholders in the superficial areas of the cortex. So one question you might ask if you want to understand their relationship to function is what if you take them offline? What if you just take the interneurons away? What happens to mice? So I'm going to cheat a little here because certainly what I'm doing is not as clean as just turning them on or off, though with recent optogenetic, chemogenetic tools, that's becoming readily possible to turn these cells on and off, and something we're really interested in doing. This is a genetic trick where we kind of remove a gene that's needed for the population and disable them. So when we do that, we're removing a gene called NKX2.1. We've only done it to about 30% of the MGE cells, and you can see this poor mouse, which now has lacking about 30% of the interneurons that it should have from that class, has dyskinesia, ataxia, and obviously frank seizures. And if you do EEGs, it's very clear that this mouse has very, very severe seizures. Now, what happens if you do the same thing but play it on the CGE cells? And here I'm cheating a little because instead of just shutting the CGE cells off, we're actually shutting off the cholinergic input to them and taking a look what happens. And so this is actually a very striking phenotype. The mouse moves just fine. It's well coordinated, but it seems to have rather a fixation with doing somersaults. And keep watching it, it keeps happening. So anyway, the point is not that I fully understand what the different interneurons doing, but that when you perturb them, even subpopulations of them, you get frank and severe dysfunction in the way the brain works. So here's the outline of the lecture as we're going to go through. I'm going to talk about how this interneuron diversity is made. So I have to say, neurobiologists, and you can see this, there was a recent Nature Neuroscience paper you may have seen that came out a few weeks ago from the Allen Institute that classifies, based on the gene expression, the different types of interneurons. There's been a previous paper in Science, and essentially this is neurobiologists butterfly collecting. How many, what types, and what kinds do you have? How many flavors? I have to say I'm more interested in where the information comes to make that diversity than I am how many. There could be thousands, there could be tens. It doesn't really matter. What we really want to understand is how each one gets programmed so it can wire into the brain correctly. And so we have a provisional model of that, which splits that up into two discrete events. What we call cardinal specification, which is really what they learn at birth, followed by definitive specification, which is what they learn on the job. And I'll explain that a little further, and I think it puts a good context out there for experimentation that we, and I hope others, will pick up on. Then I'm going to tell you 3 vignettes about interneurons. The first one is a role for genetic determination in interneuron diversity, basically asking, in cardinal specification, how much do they know in terms of what circuits they're going to wire into, and how much does that predict their adult mature function? The second thing I'm going to talk about refers to definitive specification. That's what happens after they settle in the correct position of the brain. How do they adapt to that environment and work? And what I will tell you about is good evidence that we and others have got that the early electrical activity has a so-called critical period effect so that they mature and wire properly. And I think that has a particular significance for disease because it's exactly during that time where we think that many neuropsychiatric diseases, autism included, have their etiology. And the idea is that if activity is perturbed or genes that are affected by activity are perturbed, you're going to affect the wiring of these cells. And finally, I'm going to tell you a new story, which may seem a little boutique and a little specialized, particularly for a crowd like this, but there's an underlying message that I think is really, really fascinating. I, at least, would have thought that you wire a brain by kind of setting out some connections in a loose and topographically sloppy way, and then maybe you lose activity to tighten that up and sharpen it up. And so it's an early blueprint which is refined by local activity. That's the kind of general picture I think a lot of us have. What we found when we actually tried looking at the way the wiring occurs is that there's a scaffold that's built that interneurons are involved in that gets torn down and reused, such that once you look at the adult brain, there are events that the interneuron scaffold is involved in that are completely not apparent because it's been reabsorbed into the system and repurposed for other things. But if you disturb that scaffold, brain function, at least in the context I'm telling you, is perturbed. I think the take-home message of that is hopefully going to be very important to thinking about this. You cannot look at the adult brain and infer simply by turning the clock back or thinking it's simpler what happened during development. What happens in development is unique and transient, and we've got to probe those events if we want to understand what's wrong in brains that are dysfunctional as adults. So this really takes you back to 2001 in my lab when we started doing this. What I had known since a graduate student in Canada was that if you took an embryonic brain, a human at maybe 5 to 8 weeks of age, or a mouse at 10 days of age, cracked open the top of the cortex and peered down into the lateral ventricle, that there are little progenitor zones named nicely the medial, the lateral, and the caudoganglionic eminence that are there, that disappear a few weeks later, and we didn't really know what they did. We assumed that that's where the neurons came from, but who they became or contributed to was a bit of a mystery. So in collaboration with some colleagues, Arturo Alvarez-Buya and Dan Turnbull, we took a genetically marked donor animal where all the cells were green or purple or some genetic mark that we could trace. We dissociated them, and then using ultrasound backscatter microscopy, we were able to transplant them back into an unmarked host. So essentially, by taking them and putting back in, we knew what we had taken, and then we could see where they integrated and what they became in the adult brain. And what we found, rather strikingly, is that those 3 different eminences made entirely different neuronal populations. Even if you took a chunk from one area and put it in the wrong area, they knew who they were and went and did the thing appropriate to where they came from. So this really provided good evidence that rather than being a blank slate, these neurons, even as they're being born, have a lot of information in how they're going to wire in the brain. And so here you can see in the purple here, these are all medium spiny neurons. That's the main principal cell of the striatum. They're all coming from this structure, the lateral ganglionic eminence. But you can see that there are nothing up here. So that's where all the interneurons are. But by contrast, the CGE and the MGE had a whole lot of cells up in the cortex. And you could even see, without too much anatomy, that these cells look different from these. And it turns out if you took this area and moved it over into the MGE, they still look like these. So they intrinsically know what they're going to become. And you can see that they look different. So after a whole lot of anatomy and a whole lot of investigation, we discovered that you can predict interneuron types based on sites of origin. So this is kind of a pinwheel of the interneurons. The good news is the entire palette is there. So you take these 2 structures, you can account for all the GABAergic cells anywhere in your brain, independent of whether they're in the amygdala or the hippocampus. And they either come from this green area, the CGE, or this light purple area, the MGE, and they have different flavors depending on where they came from. So this is kind of a breakdown. I think most neuroanatomists who really care about this would be appalled at the simplicity of this. They'd say there are many, many more. For instance, just looking at the cortex, the Allen Brain Institute recently claimed, based on molecular markers, that there are 23 different kinds of interneurons. If you add that to what Peter Somogyi said in the hippocampus, there's another 25. There's others in the striatum. There's at least 100 different detectable interneuron types in different structures, and probably more. And so what it became clear from the embryonic analysis is we could account for all of them in number, but not in type. So where does the information come from to make all those beautiful types in all those different areas? How do they wire into the system? So as I already said, we think it's a 2-step shuffle. An early one we call cardinal specification, in which they're determined at least to basic traits. And the kind of traits we think they're determined to are how they fire. Interneurons fire at very different rates than pyramidal cells. I'm breaking the rules, Jim. I hope you don't mind. They can fire literally 4 times as fast as a pyramidal cell, which makes them so effective at controlling activity. They connect to certain cell types. Some like connecting to other inhibitory cells. Some like are attracted to excitatory cells. And they go to different parts of those cells. Sometimes they go to all to the axon, sometimes to the dendrite, sometimes to the soma. And then they have different markers that tell them one for another. Now, definitive specification is really how they learn on the job. I'll give you evidence that an interneuron, when it's born, doesn't know what structure it's going to contribute to. It doesn't know if it's going to be in the hippocampus, the striatum, or the cortex. And since I don't think it knows that, how can it possibly know when it's born how it's supposed to wire? Because these neurons wire completely differently in these areas. So I think on the job, they get their local connectivity, their positioning, whether they're in one layer or another of the cortex, and finally their dendritic and axonal morphology, their inputs and outputs. That all gets controlled only after they find where they're going to be in the brain. So let me give you two examples of that. So this is work from Bernardo Rudi's lab, a really terrific student I've already mentioned, Robin Tremblay, and he's looked at what are called fast spiking basket cells in layer 5A versus 5B. They fire identically. They have the same habit of basically ganging up on the pyramidal cells and giving huge amounts of innervation to the soma, and they both express parvalbumin as well as many other similar markers. However, when you actually look at them, they look totally different. We're talking two cell types that literally live within 20 microns of each other in the brain. This guy has its axons and dendrites all restricted to layer 5A and gets input from this part of the thalamus, where this guy, who's just down below it, gets input from a completely different part of the thalamus and ignores 5A and basically mainly talks to layer 4. I think that's the kind of stuff they learn on the job. Let me give you another example. There's somatostatin cells. One flavor in the hippocampus is called the OLM. It sits in the Oriens layer of the hippocampus and projects down to l'actinosum moleculaire. There's Martinotti cells in the cortex, a very famous sort of feedback cell that sits either in the deep or superficial layers and talks to dendrites up in layer 1. And then there's this low threshold spiking cell down in the striatum, completely different structures. But they're all somatostatin. They all have very similar intrinsic properties. They all have this habit that if you stimulate them, they facilitate rather than depress. So they get stronger after repeated stimulation rather than weaker. And so the question is, how do these all work and wire appropriately? So I'm a geneticist kind of by training, so it seemed to make sense to go back to the beginning and ask how much did they learn based on where they come from. And so here are two hypotheses that were out there in terms of how they might go. You might imagine that a clone, so a single progenitor dividing down here in the medial ganglionic eminence, makes a whole bunch of sister cells, and they march in lockstep to take up a functional unit either in a layer or a column in the cortex or the hippocampus. Alternatively, you might say, well, lineage isn't very important, and instead they scatter all over the place and they wire according to where they are through some sort of stochastic probability and perhaps happenstance. So in order to do this, we did a logically simple but rather technically difficult experiment, which we were helped by Connie Sepka, who came up with these methods about a decade ago. We took a library of retroviruses that can go and infect the progenitor permanently. It inserts into the genome and puts in a little barcode. And we put tens of thousands of different retroviruses in there, each with a unique barcode. So that every time a progenitor gets infected, you've essentially put a digital stamp on it so that you can follow it. And every one of the daughter cells that come from that carries that digital stamp and carries it for the entirety of its life. Then you can let them scatter all over the place, see where they go, recover individual cells, and go read that digital code and see who's related to who. So when we did that, and by did, I mean Chris Mayer, a postdoc in the lab, and Rachel Bandler, a really terrific team that has done wonderful work, you can see a bunch of green cells, but you don't really know who's linearly related to who until you take the lid off and read the barcode. And so what you find here is something that you might interpret as lineage being important. These are sister cells, they're beside each other, and maybe that one up there got lost. So this at least seems that lineage might be important. However, as we went on with this, we found that sometimes we found a couple in the hippocampus and a couple in the cortex that may look to the casual observer as cells rather near each other, but in fact, since there's a sulcus between the two, they must have split up a long time ago in order to get to these two different structures. And then we saw stuff that surprised us even more, where we'd find a couple down here in the striatum and one up in the cortex, and they're linearly related. So lineage certainly did not predict where sisters went. There may be some subtle rule in there, but as a general rule, they can go into different structures even if they come from the same common progenitor. So to show this a little more quantitatively, this is basically distance in the cortex. Each one of these lines represents a single clone. And what you can see is there certainly are clusters in there, but even when there's clusters, there's this couple siblings that are up to 3 millimeters away from this. So it's really, in terms of a mouse brain, that's a huge distance. And then if you start looking in terms of ones that went across structure, now they're color-coded, you can see that it was not uncommon at all to find one progenitor that has one in the striatum, a couple in the cortex, and a bunch in the hippocampus. So in other words, lineage did not seem to predict structure. Does it predict anything? Well, we're not sure. Our bookkeeping isn't so good that we really have a thorough idea of all this. And we really wonder, does lineage predict morphology, connectivity, markers, intrinsic properties? I've already told you that cardinal specification means that they do learn stuff early. How much? We don't know. And so we're starting to do a more rigorous and more in-depth sort of analysis of this, and we're being helped by Rahul Satija, a new professor down at the New York Genome Center, and a colleague down at downtown NYU. And we're using a method where you can take individual cells and recover deep information about the genes that are expressed by that. And there's Rahul, and I think he's skulking back in the audience there, so you can ask him about this directly if you're interested. So with this, we said, well, let's start before we run out and look at them too deeply. Let's just take these progenitor zones. We know they make different cell types. What if we basically do RNA-seq on the individual progenitors? How heterogeneous, how stereotyped are they? And how much can we get individual groups that are within there? So you essentially collect all the genes from it, do principal component analysis, and using this TSNE plot, basically crunch down all that information down into 2 dimensions, and then using the first principal component analysis and nearest neighbor comparisons of gene expression, you can get the self-organizing clusters, which kind of suggests these cells here are similar, these are similar, and these are similar. And when you start looking at what genes are in which cluster, well, we know something about these genes. We know that nestin is a really good progenitor gene. DLX is a gene that really is indicative of a newborn interneuron. And ARX is something that's very important for migration and is also a disease gene. So using the kind of inferential knowledge of that, we can start guessing how these cells progress through and get these trajectories in a supervised way to start guessing how they go from progenitor to differentiated cell. We can also look at the progenitor zones across time to see how they really do. So this is combining what I just showed you, which is pseudotime, with real-time analysis. And with Rahul's considerable help, we can start using tree building algorithms, which are unsupervised, to see how the gene expression predicts these cells move and split and differentiate. Now, the beauty of that is when you find a split like this, you suddenly know that based on this, the algorithm predicts we have 2 cell types from a common cell type. And we can start asking, well, what's special about these cells and where does the information impinge on them so they can differentiate into different cell types? So this is really an exciting new direction for my lab. And I think this is going to teach us a huge amount about how molecular gene expression evolves in these cells and how they integrate and become mature cells in mature circuits. So I'm going to move from this very molecular, very single cell sort of analysis at the progenitor stage, following them through, to really the other end of the spectrum where we're looking at their definitive differentiation. We're looking at that step where they start wiring into the nervous system. And we've wondered for a long time about how and what are the cues that allow them to wire in appropriately. And as I'm suggesting here, activity seems to be really important. So that first electrical activity that impinges on those cells seems to be really important for how these cells differentiate and learn to wire appropriately. So this is just a kind of a flow chart following it through, and these are the steps they go through: neurogenesis, migration, morphological differentiation, and synaptogenesis. And here you can kind of see it visually. So there's the beautiful shapes they acquire during this phase. And then during this phase, they get the wiring, which is rather stereotyped, as many of my colleagues like Adam Kepesh, Mike Stryker, Yang Dang, and others have indicated. Is activity important? Well, you can do a simple experiment to start looking at that. And that simple experiment is to perturb activity and see what happens. And we have both gross and very fine scale tools that we can do this. So you can do what is one of our favorite molecular tricks, put in a channel like an inward rectifying potassium channel, which basically forces the activity of the cell down towards the reversal potential of potassium at -90. And basically, every time you get excitation, it's countered by this inward potassium current that keeps activity down, or you can knock it up the other way by putting a leaky sodium channel that basically depolarizes it. And so what you find when you do this in different interneurons is there seems to be some simple rules that apply to many, many different types. If this is the normal differentiation of two of my favorite ones, the somatostatin and parvalbumin ones, they become much, much simpler in their axons and dendrites if you knock down activity, and they become much, much more complex if you raise it in those cell types. You can also do that on a whole animal level by sensory deprivation or electroconvulsive shock to kind of temporarily, in a very artificial way, turn things up or down and you get the same result. So how does this work, right? So activity is shaping the way these cells form. Well, we've dug deeper on that in the last few years, and I'm going to summarize what literally is about 7 years of research down to about one slide, and a really beautiful set of studies by postdoc Natalia DeMarco, who now has her own lab at Cornell, and Theo Karianis, who will soon be starting his lab in Zurich. So this is a neurogliaform cell, one of the least understood interneurons in the brain, and we figured out a clever genetic trick where we can target it and manipulate it. And the simple thing we found when we did it was that if you reduce the activity of the cell, it withers and doesn't develop properly. It becomes just a shadow of its former self and has this very rudimentary morphology. And we started asking, okay, activity is important to the cell. What activity? Who's talking to it? How's this happening? So we were helped by a really cool trick that Ed Callaway came up with and Ian Wickersham, where they used a specialized modified rabies, where you can go in and infect the cell using genetic tricks so that you only infect a target population that you've manipulated with this rabies virus. And then over time, it transports to the cells that make monosynaptic connections with it. So by infecting this cell, we know that these cortical cells and these subplate cells connect with this cell. And surprisingly, a whole bunch of thalamic cells connect with it. So this is a lot of details of anatomy, and you might gloss over and think, why is that interesting? Well, it's interesting, among other things, because thalamus doesn't actually connect to this cell type in the adult. So this is our first hint that there's something happening developmentally that isn't true later. So, okay, it's connected up with these cells, and if I perturb that cell at this time, it withers and doesn't like it too much. Okay. Who's talking to it, and what is it saying in kind of colloquial sense? Well, we can use genetic tricks to silence the cortex by putting things like tetanus toxin in, and that basically, using a genetic trick, only shuts down this population, leaves this guy intact, and then we looked at what happened to it. Not much. It didn't seem to care. You could shut down all its cortical afferents, and it developed at least morphologically like it would normally. So then we replayed the trick, and this time we did it in the thalamus, did the same thing. And now we phenocopied beautifully what we had seen before. So there was something different about the way the thalamus was talking to this cell than the cortex was. Well, we have optogenetics tricks that we can do this. Actually, Dick Chen's lab was actually implemental with Deseroth and Ed Boyden in helping develop this stuff. So what you can do is put channel rhodopsin into these cells, excite them, and then see what this cell is saying. And when you do that, you get what you would expect, a strong glutamate response with NMDA and AMPA currents. And then we can do the same thing in the thalamus and compare it. So they don't look that different, but one thing we noticed is that the NMDA current was considerably bigger and actually much more sensitive to imfemprenol, which is indicative of a special NMDA receptor that's more calcium permeable. So we thought, that's really interesting. And we started drilling down, and this is kind of what, after a whole lot of work and a lot of genetics, what we concluded. Thalamic activity is really important for this cell to develop. It goes through an NMDA receptor that's particularly calcium permeable. It seems to signal through this calcium sensor, calcineurin, that's part of the way you get electrical signals from the surface down to the nucleus, and it affects the differentiation. But actually, we found out something even cooler. When we did that and the cell was all withered and unhappy, we basically looked at the rabies tracing and said, well, does it connect up differently? And it did. So I'm going to do that a few times. This is the normal connectivity, somewhere around 70% thalamic to 30% cortical. And when we blocked the NMDA receptor, so we blocked this signaling cascade, this cell rewired. So it's not just important for its morphology, actually the way it wires into the brain is dependent on that activity. So these are the great people who did it. Natalia, who has her own lab, Theo, who soon will have his own lab, Rashi Priya, who's just about to graduate in a few months, and Shebnam Tunç Demir, who just moved up to Renee Hen's lab. He's a lucky guy. She's fantastic, and she's going to continue her work up at Columbia. So activity links to wiring. Activity links to morphology. What's it doing? So this is something I picked up from Dick, what he would call E to T coupling. And I think it's in beautiful short form really gets you to what I think is the excitement here. Somehow activity is changing the transcriptional profile of this cell. There's a link between the two. Here's the second thing we've become interested in. How does activity change alternative splicing? You can change gene expression not just by turning a gene on and off, but by deciding which splice forms you use that in effect changes the function of the protein that comes out of it. And both of these seem to be tied in to activity. So one of the things that affects activity is this sensor. I've already mentioned it, and it's prominently mentioned in the review that I did with Caitlin and Dick, the L-type calcium channel. And it's known to be result in a syndrome when you have a mutation of this that basically makes this hyperactive. One of the transcriptional cues we became interested in is this transcription factor MEF2C. It's already known to be activity regulated. Mike Greenberg showed that a number of years ago. And we have been able to see, though I won't show you the data today, that when you get rid of this, basket cells really drop dead and they do not develop. And finally, there's this gene, which is a splice factor, which Douglas Black has shown is activity regulated, called RB-FOX. And it, too, is a gene that's associated with disease and activity regulated. And interestingly, all three of these genes have been implicated many, many times as candidate genes for autism, intellectual disability, and schizophrenia. And there's kind of a spectrum of how they're each involved in it. So I, some years ago, along with Xavier Jaglin, a postdoc in the lab, applied to the Simons Foundation to ask a question. Again, we know the genes, when mutated in humans, have a propensity to cause these diseases. How? Now, in a way, I'm going to admit it, mice geneticists cheat. I would be the first to admit it. And you know how we cheat? We look at homozygous and conditional mutants. We remove the genes wholesale from cells and see what happens. That's really good for understanding what the genes do, but it's not really good for modeling a human disease. Other than syndromic conditions such as Rett syndrome, where you have homozygous loss of it, or it's on the X chromosome, so in effect you have that. Most of the genes that affect these diseases are heterozygote mutations. So what happens in mice if you make a heterozygote mutation? Can you make an argument that there actually are endophenotypes there that you can study? Now remember, mice and humans are about 90 million years apart, and it is not a given that what happens in a human happens in a mouse. But it's certainly a good model to go look at it. And so we were, with Simon's money, we started to explore whether these mutations here, and actually heterozygote mutations in this, loss of function, cause phenotypes. So instead of showing my data here, I'm going to steal from Dick Chen's playbook and show you some beautiful data that Patrick Bader and him published a few years ago using Jackie Crawley's 3-chamber test. And that's essentially you take a mouse in its home cage and you give it a choice of playing with a new novel mouse or very little. Now, most mice, being the social enterprising lads they are, will pay attention to the new mouse. But a Timothy syndrome mouse, as Patrick beautifully showed with Dick, spends its time preferentially attending to the empty chamber or the one filled with blocks. And you can see this statistically when you do that sort of analysis. So a mouse with the same mutation as exists in humans of this, in this L-type calcium channel has social behaviors reminiscent of what happens in humans. So that may not say it's the same thing, but it does validate the mouse model of that as a way of studying autism. I'm going to show you some recent work, and this is Patrick Batter, happy as always, and Dick Chen. This is some recent work that we've done in collaboration with Adam Marr, who runs a beautiful, really a state-of-the-art behavioral core at NYU. And he started to digitize this. These analysis, I want to emphasize, were done blind. We did not know the genotype. Begonia and him did the three-chamber, did open field conditioning with this. And what you can see here is this is what the normal wild-type mouse would be. And you can see the MEF2C mouse preferentially sticks around in corners and doesn't like the middle and doesn't like crossing it, and really, really shows signs of anxiety. Another measure of anxiety is this dark and light area. You basically bait them out here with food and curiosity, but if they're nervous and anxiety-ridden, they tend to like hiding in the dark. And the MEF2C hat clearly shows a preference from dark versus light spaces. So this at least is consistent with the phenotype there. And this work was done actually very recently by Begonia and Adam. They also did the three-chamber test. You're already familiar with it. I didn't show you a control, but this is what a control should look like. There's the bait of the new novel mouse, and you can see wild-type mice greatly prefer the investigation of a new playmate and ignore the empty area, and the MEF2C does exactly the opposite. I'm just going to show you a tidbit on the RB fox. We recently only got the heterozygotes, so we've only been able to look at their ultrasound vocalizations. But there's a really striking phenotype, and it's that by P14, when you separate a RB fox het from its mother, it makes 3 times the number of frequency of vocalizations as does a wild-type mouse. And while this is still early days, it is indicative of an autism-like phenotype. Both Rett mice as well as BTB-R mice, a genetic model of autism, show the same sort of vocalization. So we're hopeful that this is going to start showing us that these models really as hets have validity in investigating mice as a model of it. And what we're going to do next is remove these genes just from our favorite cell types, the interneurons or the excitatory cells, and ask if we phenocopy it. And so that way, I think we'll get some direct evidence if the same mutation in select cell types can phenocopy the kind of disorder you see in humans. So I think this is an important next step to go from genes to behavior and understanding how these genes function. So I'm going to go from there to something a little closer to what my lab does every day, and that's a more molecular analysis of this. And in terms of our alternative splicing, we really have become interested in how it's regulated by activity, and particularly how RB fox might play a role in it. So the simple evidence that splicing is controlled by activity in interneurons was done by a very simple experiment. We gave electroconvulsive shock to a mouse. We collected all the interneurons, and then we looked 3 hours later to see if the level of splicing had changed, and it goes up about 3-fold. So something as simple as just globally raising the activity in interneurons changes the amount of splicing tremendously. So the question became, who's controlling it and how is that done? And so we became very interested in splice factors that might be activity regulated, and we centered on RB fox. We did that because work by a number of groups, Dan Geschwin, Bob Darnell, and Douglas Black, have all indicated that RB fox has a higher mutation concurrence in patients with psychiatric disease. And here's just a list of everyone from Jonathan Sabat and Michael Wigler to Dan Geschwin to Carroll and Cook, all showing in a variety of circumstances that mutations in RB fox are comorbid with a variety of psychiatric diseases, autism included. So Yeah, I think this didn't come out quite right. So what that's supposed to show you but doesn't, you can imagine bars here. Close your eyes, think. Basically, it's enriched in the PV and somatostatin interneurons and far less in this population. So it's a pretty simple bar graph. So it's in the population we were interested in, and these two have been investigating what it does. So really, the hypothesis is that RB-FOX in an activity-dependent way is somehow involved in the maturation of these two populations where it's highly enriched. And so we do what we often do, which is remove RB-FOX from our population selectively. And we had a hint it was going to work because Douglas Black beat us to it. But he used a Nestin promoter. That's basically generic neuronal removal. And he got animals with epilepsy, which is always indicative of something you might expect in an animal where interneurons aren't functioning. So we've taken our favorite interneuron drivers to remove them more selectively. I won't go to the details, but it's a way of removing it temporally and in specific interneurons and see what happens. So what we found was wild-type mice, not surprisingly, survive very well. Ones in which we removed it globally in interneurons were fine to about P18, and then they start dropping until there's almost complete lethality. That's kind of what happens by about P30. So the animals don't like it when you remove RB-FOX from interneurons. The other thing you see is they're much more susceptible to seizures. So if you kind of predispose them to seizures pharmacologically, you can see you can induce a seizure in an RB-FOX conditional interneuron knockout much easier than you can do in a wild type. That usually means you've lost interneurons. We've seen it many times. So we kind of expected we'd go in and RB-FOX would kill off a bunch of interneurons. So we were surprised when we went in and looked that we found that there were 20 to 30% more interneurons in all layers than we expected. We got more interneurons when we removed RB-FOX, not less. So this kind of suggested a paradox. On one hand, you have higher densities of interneurons, but on the other hand, you have seizures, lethality, and poor survival. How come? You got more interneurons. What's the problem? Well, the idea is that yes, you have more, but they're not functioning properly. And we were basically immediately thought of a paper that my friend and colleague Arturo Alvarez-Buya out in San Francisco had done, which showed that interneurons undergo a huge swath of cell death right over the period when we thought RB-FOX was functioning. And what it looked like here is that without RB-FOX there, the cells don't die, but neither do they mature. And this is pretty typical. Activity-regulated cell death is a way you prune out the guys you don't want by basically getting them online where they start sucking up energy like wild men, and you basically only let the ones that are doing the right thing survive. If you prevent their activity, you prevent the cell death. So that's what looked like was happening. So we wanted to drill down and look at the connectivity of these cells and see if it was changed when we removed RB-FOX. So there's a set of genetic tools here, probably not too important to know the details unless you're an aficionado. And I'll kind of go through it quickly, but basically this is a combination of physiology, immunohistochemistry, basically looking at inhibitory synapses presynaptically by this marker synaptotagmin 2 that in basket cells marks presynaptic GABAergic input from the PV cell onto the pyramidal neuron, and Gefrin, which is a marker postsynaptically. So when the green and red goes together, you know you have an inhibitory synapse from a basket cell. It's that simple. So the first thing we did was look at the physiology, and you see that even before you look at the connectivity, the mutants don't fire nearly as well. This is a really reliable, robust inhibitory cell that matures. It fires at these incredible rates. It has low failure rates. And that doesn't work so well when it doesn't have RB-FOX. It looks immature, exactly how we hypothesized. Then when we looked at these pyramidal cells, and you can see them here, and you can see them nicely decorated with those green and red dots I promised, and then looked at a mutant, well, a lot of them were missing. If you quantitate it, as Bree did, you can clearly get about 10% of the normal inhibition from the basket cell onto these cells. So RB-FOX is needed for the basket cell to target the cells they like to target, the pyramidal cell. Okay, they don't mature. What about the Martinati cell? That's the other cell we saw with a lot of it. using other genetic tricks, basically the same logic where we label in green the presynapse and red the postsynapse. We went in and looked at it. This is the normal pattern you'd see. I mentioned it quickly that the Martinati cells basically innervate the superficial part of cortex, but sit down here. And so if you look at the synapses of these Martinati cells, they're right here in the superficial cortex as you would expect. This was a shocker. They wire with everyone. They basically don't care who they connect to. They're now wiring in all layers, in all places. And so it looks like they've become the superhero of inhibition and gone everywhere, but not so quick. That shows they wire there. And even though I'm not showing you the complete data, because there's a postsynaptic marker here, Gefrin, you can see they line up. So we think they're real synapses, but we don't know if they're efficient. It may be that instead of concentrating all their effort where they should be, up in the superficial layer, they're making a whole bunch of weak synapses everywhere. And so even though they're making more synapses, they're less effective. So that's kind of where that story is. We know that loss of function causes loss of inhibition. PV cells have reduced synaptic outputs. Somatostatin cells have increased synaptic outputs, but perhaps weaker function. And now, through a combination of RNA-seq to analyze all the different changes in splicing in these two cells, combined with physiology, we're digging deep and looking at it. And while that will give us good granular detail about these cells, we think the implication is that this may explain some of the symptoms of the patients who lack this gene and ultimately have psychiatric disease. So in the last few minutes, I'm going to tell you one little vignette. It's really one that's probably more geared towards people who are interested in physiology or function. But I think there's a real simple take-home that's really, really powerful. And that is developmental scaffolds in the brain are transient and essential to the wiring of the brain. So for anyone who wants to understand what's happening in a psychiatric patient, a schizophrenic, ADHD, obsessive compulsive, autism brain, it's not enough to get the most detailed analysis of the mature animal. You've got to know what's happening as it develops. These genes are clearly ones that act during development, and the etiology of many of these diseases happen during development. And I suspect there are events there that you have to catch in the act or you miss them. Yes?
You don't have access to genetic seed that extrapolate from behavior from a young age through on development?
I think the guess is with enough quantitative analysis, quantitative behavioral analysis, which is what Adam and Begonia are really trying to do. That is the hope. I don't think that's been fully played out. But that, certainly if the disease is there early on, you would expect that if you have the right sort of analysis, maybe it'll come out as behavior. Maybe you need to image brain activity. Maybe there is a bioassay you could get simply by taking a blood test. But I think through those parametric analyses, we ultimately hope that you will be able to do that. But that's a promise rather than a reality. Any other questions? Okay, we can save them to the end. So you've already seen this game. This is where we take the rabies and we mark it. This time we're doing it to a somatostatin cell because we're interested in that population developmentally. We're interested in that because they're the earliest born interneurons. And when we looked at the wiring, just as we did with a neurogliaform cell, we see this huge thalamic input, which again, we don't expect to see in the adult. So that was a surprise. And when we compared it, this is the adult wiring of that somatostatin cell, and you can see it gets a whole lot more cortical projections and a whole lot less thalamic projections. And if you quantify it, you can see various things change, but the big difference is this early thalamic projection seems to disappear. And it does it like clockwork. It doesn't like gradually fade away. It's there, it's gangbusters, it's there till postnatal day 8, and then by postnatal day 12, it drops like a stone. So really, there's this 2-day period in a mouse where it goes from being heavily wired with thalamic inputs onto the somatostatin cell to almost none. So in order to make sure, rabies is kind of a new method. People are kind of not sure they trust it. So we did this trick using the ultrasound to just label up the thalamus with lots of channel rhodopsin, and then we went in and recorded from the somatostatin cell to see how much input it was getting. We used the pyramidal neuron, who also gets input from the thalamus, for comparison, and this is what we saw. So at P6, big whopping current onto the somatostatin cell, almost nothing onto the pyramidal neuron. P9, it's reversed. That's the somatostatin neuron, that's the pyramidal neuron. Wait till P18, wow, completely changed. So the anatomy matches the physiology. It gets a big input, it loses that big input. So who's it talking to? You know, basically a connection occurs and disappears. If it's doing nothing, who cares? Well, so we went in and looked at it. We did the same trick by putting channelrhodopsin into the somatostatin neuron and then recorded from the PV and the pyramidal neuron to see if it's talking to those guys. And indeed, developmentally, it's talking about equally to the pyramidal neuron and the PV interneuron. But by P15, this is fading away and this is getting stronger, and that trend continues. So basically, this is what you got. A thalamic projection going to this cell, and then this cell talking to the pyramidal neuron and the PV neuron. And then after P9, this projection's gone. This guy now projects strongly to this and this. This is a classic, beautiful circuit that Barry Connors and others have described. It basically provides feed-forward inhibition. So let's just play neural circuit for a minute. Sensory information comes in, sparks up the pyramidal neuron and says a whisker's moved, there's sensory information to attend to, but it also talks to the PV neuron. This guy fires, and then a moment later, almost simultaneously, this guy fires, and then he shuts this guy up. So think of it as a computation. If the PV cell wasn't there, it would go. With the PV neuron, you get a nice little. So essentially, through feed-forward inhibition, this guy's making sure that the signaling to this guy is precise and well-timed. That's what you ultimately want. So, does this matter for this? Maybe it doesn't. So we decided to test that. And we did that through kind of a brute force method. We simply went in developmentally and killed off about half of these cells using diphtheria toxin. You can see it here. There's about numerically half the number there. It's a genetic trick. And when we did that, we then said, okay, let's listen to the PV cell and stimulate from the thalamus. And this is what you should get. This is what you do get. So without the somatostatin neuron talking to the PV cell developmentally, the PV cell never gets that strong thalamic input. And instead of beautiful, you have this long driving firing of the pyramidal neuron. I'm not showing you, but that is actually what happens because feed-forward inhibition isn't there. Hey, the somatostatin neurons still are dead after P9. Maybe I'm just fooling you, and it's really something it's doing later on that's important. So we played the same game, but we did the exact same experiment after P9. We killed off about half the somatostatin neurons and then looked at the same thing, no change. So that transient connection, that connection that was there and disappeared, is vitally important for the wiring of the adult brain. Now maybe that's just a boutique single example and it doesn't generalize. But my suspicion is it does. And I think as we look deeper into the way the connections of these interneurons form and change, we're going to discover that there's a whole world of scaffolding going on that's vitally important for our adult brain to work properly. And so just to give credit where credit's due, this was the graduate work of Shebnem Tunçdemir, helped by another graduate student, Bree Wamsley. So this is just an outline of what I told you. I started off by talking about lineage in interneurons and how lineage predicts certain basic types but doesn't predict their position or wiring in the brain. We went through some role of activity and how activity and activity-regulated genes are important for the way the cells mature, their axons and dendrites, and form connections. And finally, I told you a scaffolding story of how interneurons make transient connections important for their adult connectivity. And so to end off with a picture of New York, which is kind of appropriate given our snowstorm last week, I'm giving all the names of the lab on a background of New York. And I won't go through the names because I've really given credit as I went through, but these are many, many people who have been implemental to this work. Thank you very much.
Thank you, Gore.